Hello, welcome to the EASL Journal Club. My name is Jesus Bañales. I am member of the EASL Educational Committee, and I will serve as moderator of this webinar. In today's episode, we will discuss the manuscript molecular-based targeted therapies in patients with hepatocellular carcinoma and hepato-cholangiocarcinoma refractory to atezolizumab plus bevacizumab. This is a paper recently published in Journal of Hepatology, and we are lucky to have the first author and presenting author, Lendi Limousin, then the senior author, Professor Jean-Charles Nolte, and also two experts in the field, Professor R. Vogel, who will act as challenger, and also Dr. Pedro Rodriguez, an EASL Joint Investigator Task Force member. Welcome, all of you. Next slide, please. So just some reminders for those who are attending the Journal Club, please use the Q&A chat or raise your hand if you would like to ask questions orally, and please turn your camera on. For those who are active on Twitter, please use the hashtag EASL Journal Club if you want to ask questions or share your thoughts through X. And also just remind that the EASL Journal Club will be recorded for the purposes of on-demand viewing, and the recording will be hosted in the EASL campus. So now the floor is yours, Wendy. Thank you.
Thank you. Thank you for inviting us. I do not seem to have control. Yes, I do. HCC molecular landscape has been extensively described, and the main genetic alteration involves third promoter, TP53, IB1, CTNNB1, APC, and AXIN1, but also genes from the oxidative stress pathway and from the mTOR MAP kinase pathway. Hepato-cholangiocarcinoma shares molecular alteration that are common to HCC and also to cholangiocarcinoma. The 2025 French Medicine Genomic program is a nationwide initiative, and it has 3 indications in oncology. It's rare tumors, cancers of unknown origin, and cancer refractory to systemic treatment. Whole genome, whole exome, and RNA sequencing are performed on frozen tumor samples, and all results are discussed in the molecular tumor board. If an actionable genetic alteration is identified, then targeted therapy is recommended. For this study, we retrospectively collected data from 1 tertiary center in France between September 2020 and January 2023. All patients with HCC or hepato-cholangiocarcinoma progressing on atezolizumab with a good liver function, a good performance status, and frozen tumor samples available were discussed in the first molecular tumor board. Then frozen tumor samples and blood samples were sent out for sequencing, and patients were treated with TKI. We're waiting for results. When results will be available, then they were discussed in the second molecular tumor board, and it led either to targeted therapies or another TKI. So we had 135 patients treated with atezolizumab during this period of time. 100 experienced a progressive disease, and 34 were fit for second-line therapy. Finally, 20 patients had genomic analysis performed and were included. Among the 20 patients, we had 16 HCC, 4 hepato-cholangiocarcinoma. 70% were male, median age was 57. 50% had a chronic hepatitis B infection, and 50% had cirrhosis. 60% had only 1 line of systemic treatment, being atezolizumab, and the other 45% had up to 4 different lines of systemic treatment. They were mostly BCLCC, mostly metastatic, and genomic analysis were performed on biopsy for 19 patients and on a surgical sample for the other 1. 10 tumors were collected before atezolizumab and 10 after atezolizumab, and the median time from sample reception to result was 62 days. Genomic analysis results were analyzable for 19 patients, and we represented on your right part of the screen the main genetic alteration with TP53, RB1, third promoter, CCND1, and FGF19 amplification. The median number of somatic non-synonymous SNV was 19.5, and mutational signatures were identified, such as SBS24 in 5 patients who were born in Africa, and SBS22 in 1 patient who was born in China. The median homologous recombination score was 25, and 1 patient had a high score of 88. 14 patients had at least one actionable genomic alteration for a total of 20 actionable genomic alteration identified. We classified them according to the SCAT classification, and 30% were SCAT3, 60% were SCAT4. Among the 14 patients, 9 were treated. So we had 3 patients who were treated by everolimus matched to a PTEN TSC1 or TSC2 mutation. The patient with the TSC2 mutation had a complete response, while the other 2 had a progressive disease. 1 patient with a high HRD score and a ERBB2 overexpression was treated with trastuzumab plus niraparib, resulting in a stable disease. 3 patients with FGF19 amplification were treated with trametinib, resulting in a progressive disease. 1 patient with a focal CDK4 amplification was treated with palbociclib, resulting in a partial response. And finally, 1 patient with a high HRD score and a SBS3 mutational signature that is associated with defective homologous base repair system was treated with olaparib, resulting in a progressive disease. So then we wanted to show you the 2 patients with a radiological response. The first one is a 48-year-old female patient with a metastatic hepato-cholangiocarcinoma. After progressing on sorafenib, atezolizumab, regorafenib, she was treated with palbociclib matched to a CDK4 amplification, and it resulted in a partial response that lasted 16 months. The second patient is a 68-year-old female patient with HCC that developed on a cirrhotic liver. She had experienced progressive disease with atezolizumab, with sorafenib, and was then treated with everolimus matched to a TSC2 inactivating biallelic mutation. It resulted in a complete response, and she's still under treatment after 12 months of treatment. So with this work, we wanted to show that molecular-based target therapy is feasible in patients with HCC and hepato-cholangiocarcinoma who are progressing on atezolizumab, and it can be useful in a subset of patients. The main limitations are that frozen materials are required. There's only a limited number of patients, so we need more validation studies, and the treatment strongly depends on drug availability. Thank you.
Thank you very much, Wendy, for this presentation. So next we will have a poll. So everybody that are connected could answer to this poll. So the question about the genetic landscape of HCC, what are the true answers? The most frequent genetic alterations that are TP53, TERT, and beta-catenin are undruggable. FGFR2 fusion are identified in 5% of the cases and could be treated by FGFR inhibitors. Microsatellite instability is observed in 10% of the cases that could be biomarker predictive of sensitivity to immunotherapy. FGFR4 inhibitor have been tested in HCC with FGF19 amplification. And finally, KRAS mutation are observed in a subset of the cases, 5 to 10, and could be treated using specific KRAS inhibitor. So you can answer, and we will wait 10 to 20 seconds to have the answer. So Paul, one question. There is multiple choice, and you could have more than one good answer.
And then it's time for discussion. Now, please, Professor Vogel, Pedro, please go ahead.
Okay, so should we start with the paper or with the poll?
No, we go to the discussion of the paper. The
Yeah.
poll, we can discuss it later, yeah. And
Okay.
see
Okay, perfect. Yeah, I mean, thank you very much. I think it's interesting. And of course, I mean, so my role is a little bit the challenger. So I have to ask you some critical questions. And so just, I mean, like looking at this from like top to bottom, is it a good result or a bad result? I mean, you started with more than 100 patients, right? And then you were able to get druggable alterations in 20, and the number of patients that really responded to the treatment are in single digit. So is it worth all the effort?
I can answer to this question. So it's a good question because in terms of time it takes, money, and so the first point is I think we don't need to put the results as a result in first line. It is in second line. So in second line, we know that at least in clinical practice, 30 to 40% maximum of patients could have access to a second line treatment. So I think we could take it as a second line or maybe also third line result. So it's not 20 patients among 100, it's 20 patients among 45 and 40. So clearly, it's only a subset of patients who could have access to a treatment in second line. In clinical practice in my center, but in most of the centers, only 1/3, 30 to 40%. And in this 30%, half of them have been included in this analysis. So I think it is a strategy for patients that have no other option currently, because we have no clinical trial after the first line in our center. Most of the patients have been treated by tyrosine kinase inhibitors. So it is a third line proposal. So it's not so bad, I think, in third line. And you can see that some of patients with response have received 3 lines of treatment or 4 lines of treatment. So I fully agree with you. It's not for every patient. It's only for patients resistant to immunotherapy, and most of them have received tyrosine kinase inhibitors. So it's a third line proposal. It's a proof of concept. But I agree with you. It's one of the limitations.
Okay, so next question. Is it really for patients in second, third, fourth line? I mean, should we really wait so long, given that for many drugs, we do not really have good clinical trials? Evidence is low. We need to argue with health insurance companies, maybe. So for you, I think it took 2 months to get the results. So do we really have the time in HCC and BTC to wait after all evidence-based therapies have failed, or should we do it upfront?
It's not a validated treatment, molecular-based therapy in HCC. But whatever, why half of the people don't receive any treatment, even if we identified a targetable genetic alteration, because they progressed very fast. And they are not. They don't have access to this treatment because performance stage 2 and liver failure. So I think it's like in biliary tract cancer. Now the guideline is to move very fast to sequencing. In HCC, it's more debatable, but I fully agree that the issue is liver failure after progression. So maybe in the future, in 10 years, everybody will be sequenced because it will be less expensive. It will be faster. And we will have access directly in second line to this potential treatment. But it's only wishful thinking. Maybe you can have a comment about biliary tract cancer, Jesus, about sequencing. If you need to do sequencing first, very fast for biliary tract cancer.
Yeah, even the accessibility to tissue, it can be solved with the new good results on liquid biopsy, and I think these data are very encouraging, particularly the complete response, of that patient to everolimus. This is very incredible, I think it's a very, very important result. Yeah.
So maybe the third question. So what about, so what you're doing is whole genome, whole exome sequencing, transcriptome. Is it a little bit over the top? I mean, so we do know that the druggable alteration in HCC, we know that most of them, as we just asked in the poll, are not druggable. So do we need these excessive sequencing? Is this academic research, or is it really patient care as of today?
So the first answer is when we used our usual genomic test in clinical practice, we missed more than half of the druggable genetic alteration in HCC. The second point, I think it's a question of future. I'm sure that in 10 years, everybody will have it will not be panel sequencing. It will be whole genome, whole exome sequencing and RNA-seq, because everybody moves very fast. The analysis will be very fast, more easy. So I think in 10 years, nobody will ask, I want to do a target gene sequencing. In my opinion, because it's a way how genomic move. It's not target gene sequencing. It will be whole exome, RNA-seq for tumors, and we will discuss after, because it will be cheaper, it will be faster, and it will be easier to analyze. So I think currently it's a good question, because only I think 10 genes, 15 genes are potentially degradable in HCC, mutated in less than 2 to 3%. But I think it's a question of the future of genomic, and in 10 years, it will be whole exome, whole genome, RNA-seq, and I'm sure of that. But it's only a discussion about the future.
Okay, so the next question: how much evidence do we need to use a specific treatment in HCC? I mean, so when we take the example that Jesus also just mentioned with the mTOR inhibitor, right? I think it's a very nice example, because I think, I mean, if you would have asked me up front, I would have predicted that one may respond, right? But you treated 3. So how much evidence do you need to really start this kind of treatment?
So yeah, it's there is two answer to this question. I think we do it because there are very few options for this patient. It was they experienced 2 tyrosine kinase inhibitors and 1 immunotherapy, and no clinical trial available. So it's not a very good scientific based answer. I agree, but it's like that. Second point is very interesting in terms of 3 patients with the mTOR inhibitor, only 1 response, and it's more difficult than we think, because probably P10 deletion is not the same as TSC1 and TSC2 mutation, first. The second one, the thing that we don't assess very well in this analysis is clonal mutation versus subclonal mutation, and maybe some of this mutation could be subclonal. And currently it's not very well analyzed in the French Medicine Genomic program, because we put all the potential driver among 10% of VAF. Even it's not the same to have 10 versus 40% of VAF. So maybe it's a question of type of mutation in the mTOR pathway. Second, a question of clonal versus subclonal mutation. Third point, it's a thing that is very difficult, but you know it's the same in cholangiocarcinoma, is a co-occurrence between driver mutation. Sometimes you have 2 driver mutations at the same time, and maybe some of the clue to understand the answer to a systemic treatment associated with druggable genetic alteration is to understand what are the association between each driver. And currently we have not enough data to answer this question. But I think it's a very important question in terms of what are the combination of driver mutation together.
So I think it's also important when we think about monotherapy. I think with mTOR inhibitors, we do have at least some experience from the transplant setting, right? So we know a little bit about the side effects, but still, I mean, specifically when we think about co-alterations and combination of maybe different targeted therapies, toxicity comes also into place, right? And usually toxicity is not really very well documented and reported, right? So and therefore I think it's an important question as of today, to which extent we can extrapolate data from other tumor types to our specific GI tumors, and specifically hepatocellular carcinoma, right? So and therefore maybe again, so I mean, one other example is basically the FGF19 amplification, right? Where you could argue this would be a good argument for an FGFR inhibitor like lenvatinib. On the other hand, I mean, I think you have used a MEK inhibitor. So how much, I mean, not for your patients, but like looking forward in the future. So what would be, also based on your experience you have just published, so how much evidence do you require today to start really a treatment?
So the first point, I think it's experience in other type of tumors. I'm for sure. Second point is if we know that it's a true driver in HCC, sometimes you identify the mutation in the genes, the functionality is unknown. And if we do a molecular tumor board and we go deeply in the analysis of a whole genome sequencing and whole exome sequencing, sometimes we don't know the functionality of substitution. And if the functionality is unknown, if the driver genes have never been identified in HCC before, I will not go to use a drug against this genetic alteration. And also, I will use a drug that have shown efficacy against a genetic alteration in other type of tumor at least. and acceptable safety. And I agree that it's one of the limitations for cirrhotic patients. And just to give you some example, there is one patient with olaparib that have been treated in this study that have been neutropenia, thrombocytopenia, and so on. And we need to stop the treatment due to this adverse event. And maybe it's because he has a chronic liver disease that increase the risk of toxicity. We don't know, but we need to be careful also on this potential toxicity, and because we use also the drug off-label, and so we need to inform the patient of this. It's outside any clinical trial. It is because only there is no evidence-based medicine in third line, in fourth line, in HCC that we propose such therapy. And I want to be clear on that. It's clear on that. And if we have a clinical trial, we propose a clinical trial.
So, Jean-Charles, so based on your comments and a little bit on the design, would you recommend to move forward with those mutations, scale as SCAD3 and no SCAD4 or both? What do you think, considering the SCAD scale of ESMO?
So maybe, Wendy, you can discuss about SCAD, but I think most of them are SCAD3 that are
Yes.
with response. Most of
So some of them are SCAD3, right?
Yeah.
Based on other efficacy.
Genetic alteration associated with response are SCAD3 in my, if I remember well. Can you confirm, Wendy? Because a lot of SCAD4, because we don't know how to treat them, and SCAD3, most of them are SCAD3 except maybe the palbociclib one, but
TSC2 is SCAD3,
yeah.
ERBB2 and high HRD with the stable disease is also SCAD3, and the last one, the CDK4 amplification is SCAD4.
Yeah.
Experimental evidence. Okay, good. Okay.
And I would like also to highlight, just maybe I could do the job of Anne to highlight the limitation. So it is, we include hepato-cholangiocarcinoma, and maybe we could discuss a little bit because it's very interesting, but because for this rare tumor, because maybe it's interesting to move to this genomic-based target therapy because there is no evidence-based data. And we find something sometimes very strange genetic alteration. So it is not very, very well known in terms of genomic landscape. Even if there is publication, for example, we have a KRAS amplification. KRAS amplification is not very frequent, except even in cholangiocarcinoma. And in hepato-cholangiocarcinoma, we found CDK4 amplification, KRAS amplification. So maybe it's a good target also. So, rare cancer, nothing very, very well known to be efficient in terms of systemic treatment. And so maybe it could be also a good target for sequencing. Just a general comment.
Pedro has the hand raised.
Yes, first of all, congratulations on this excellent work. And my question goes more in the direction that in what you are talking about, the hepatocholangio tumors. Do you have any recommendation on the use of targeted therapies for this subset of patients? Because it is more or less known that these tumors are kind of different from HCC, are kind of different from cholangio. They are kind of a distant entity. And they have, as you mentioned, a different mutational burden and different mutations that might impact on the response to targeted therapies. So should they be managed in the same way as HCC or cholangio, or should we be careful and manage these patients differently from the other two types of tumors?
For the clonality, it's true, different.
Yes.
I think we don't know. Just to be clear. But we don't know. What we can propose is not evidence-based. There is first, diagnosis is very difficult. Diagnosis is very difficult. And even if you ask two pathologists to review a slide, most of them said it's a HCC, it's a cholangiocar, I don't know, maybe it's a hepato-cholangiocar. First, the diagnosis is difficult. The second point, there is no data about systemic treatment prospective. There is only retrospective data. And third point, I think it's very rare, so we don't know. So it's boundaries between research and clinical practice for me. It's one example. Very rare cancer, so we could do genomic testing for all these patients, only very few patients, and discuss in multidisciplinary tumor board what to do. And at the end it will be at the boundary because between research and clinical practice, because clinical practice is based on nothing. So I suggest to do sequencing. Maybe not whole genome and so on, but at least target sequencing for these rare tumors and discuss the management in our multidisciplinary tumor board. It's my advice, but because if not, we do treatment of HCC and after treatment of cholangiocarcinoma in first line and second line or vice versa. So yeah, I don't know, what do you think, Arndt? But
yeah, I mean, I fully agree. I mean, and I think maybe one brief comment on the how to perform sequencing, right? So I think, I mean, it's great what you are able to do in France, right? I mean, whole exome, whole genome plus transcriptome. I think this is basically like a vision for the future, right? And I mean, as you said, maybe in 10 years, we are also in other European countries, we are at that point. I mean, I think it's really great. Yeah, it's mostly academic at the moment. Yeah, but on the other hand, you get really a comprehensive picture, right? And I think when we are talking about panel sequence, there's always the danger that we use a panel that does not detect all alterations we are interested in, right? And specifically when we talk about cholangiocarcinoma and also the mixed tumors, we have these FGFR2 fusions, right? And they are missed in many panels, yeah? And I've actually seen a couple of patients with FGFR2 in-frame deletions and fusions. So I think here it's really important to use more comprehensive profiling, which also includes these rare fusions. Yeah, so I mean, if you have a very broad sequencing, it's great. But specifically in the mixed tumors, I think it's really important to look at to use a panel that allows detection of fusions.
Yeah, yeah. And what are your feedback about circulating tumor DNA? Do you think it could be an option for hepato-cholangiocarcinoma, for example, or it's nothing interesting?
No, I think it will definitely be an option, right? And I think, I mean, first of all, we have tissue, which can be challenging. We have blood, which is okay, and we have bile, right? I mean, for us as hepatologists, it's also something we should not forget. We can also use bile. It's, I mean, there are some papers out there, one in Gut recently published, but also a couple of others, and we have also started on that. So bile is an excellent source for NGS to perform NGS. And I mean, I don't know whether you have seen it, but there was like a couple of weeks ago a paper in Science, and they basically asked the question, so how can we improve detection of ctDNA in the blood, right? And they asked the question, what happens with the ctDNA? And I didn't know. I mean, do you know what's happening with the ctDNA?
They disappeared, but we don't know why. They disappeared.
Disappeared, but it's taken up by
Macrophages in the liver. It is.
Right, it's taken up by Kupffer cells, right? And they basically blocked the Kupffer cells by nanoparticles and antibodies, and they improved the detection rate by more than 10-fold in mice, right? So it could be like a contrast-enhancing agent in imaging, right? So when we have these booster, which allows detection of ctDNA in the blood at a higher rate, I think it could be extremely useful, and then we probably can do it much more and also sequentially over time.
Yeah, and just to add something, we present the French Medicine Genomic program. It was based on frozen tumor sample, and now the next step is to move to paraffin tumors and plasma. So we are going to begin to do it, but in very specific cases first, because to be sure that we could have results, but we are the first H-CCK in paraffin will be sequenced with whole exome and RNA-seq, 3' RNA-seq, and they are going to do plasma sample for lung cancer, breast cancer outside the field of H-CCK. But my personal experience with circulating tumor DNA for H-CCK, it's I agree that it's not when you go to small tumors, it's difficult to detect circulating tumor DNA for H-CCK. And I don't know if it is because of what you say about Kupfer cell and so on, but it was a very interesting paper. And maybe it's why it's sometimes difficult to detect small tumor with circulating tumor DNA in HCC. I have not the experience of cholangiocarcinoma, but for HCC, it's sure that you need a huge tumor burden to have a good detection of circulating tumor DNA, at least using the technique that we have currently.
Also take into account there is a paper published in Clinical Cancer Research in 2019 that profiled the ctDNA of 206 patients with HCC with a 70-partner gene, and they found druggable alteration in 57% of the patients. So, I mean, your study is very comprehensive with this kind of data, and I think this will evolve very rapidly, and the data are very, very promising. So, well, first of all, thank you very much congratulations for the excellent study. Thank you very much for all the very nice discussion. We can now proceed with the 2 polls. We can see the question. Jean-Charles, you want to raise it?
Yeah. So the good answer is the most frequent genetic alteration are undruggable. For sure, if in 10 years we have a good beta-catenin inhibitors, the landscape will be modified. Currently, it's not the case. And the other good answer is the FGFR4 inhibitor have been tested in HCC with FGF19 amplification. So maybe we could to move to the last. And maybe it's a question, maybe not for a poll, but we could discuss what everybody think. All the patients with advanced HCC will have a whole genome sequencing RNA-seq. Only patients with third line, fourth line will have access to such huge genomic sequencing. Only if there is drug directed against the main genetic alteration, for example, a beta-catenin, or you don't see a role for molecular-based target therapy. Maybe you could ask as a poll, but maybe we could have a round between all of us. Personally, I think it's not for HCC, but every cancer will be sequenced in 10 years, but probably using a whole genome. But we could ask for the poll, and maybe everybody can say some words. No, what do you think, Jesus, for HCC or cholangiocarcinoma? What are your thoughts?
Yeah, I think we need more evidence. This is just a very comprehensive and nice study, showing that all this genomic profiling, all the subtypings that you and others have done in HCC, and it has been done also in cholangiocarcinoma, makes sense in the search of targeted therapies based on pathway alterations. But definitely we need, of course, validation. We need to define the panels, to do the molecular profiling, to see cost-effective strategies, and based on evidence, of course. But I think this is very, very interesting, and I envision a future, a 5-year future, very, very alienated with this. I remember when we and others were talking on targeted therapies in cholangio, when the first trial were done in FGFR receptor, the people who most of the people say that this was gonna be the case. It's impossible. And now it's standard of care. So I think the future is bright in this direction.
Yes, I mean, we have a very good understanding of the genetic alterations today, right? So, I mean, a couple of years ago, it was not that clear, but now with all these comprehensive approaches and, I mean, whole exome, whole genome, transcriptome. So I think what you're doing in France clearly will add to the evidence. And in the end, it comes down that we need drugs, right? So, and this probably drives much of what we should do and what we should broadly recommend. I mean, I agree. If we don't have any options, I mean, if we have the opportunity, we take everything what we can. But on the other hand, it's also good to have like a little bit more data and a little bit more evidence. And I mean, we talked a little bit about the toxicity. So I think for p53, for example, or beta-catenin, I mean, if we really get new drugs to target these alterations, I mean, this could be a major breakthrough. And in cholangio, I mean, we were lucky, right? Because there are so many druggable alterations, even today. And in HCC, the majority is simply not yet druggable. But when we get these drugs, I mean, there's clearly a role for sequencing.
And it's important also what Jean-Charles mentioned. We need good molecular biology demonstrated that these genetic alterations are drivers of the disease progression and development in order to move forward with the next testing with drugs. This is key to identify the genetic alteration, evaluate the biological role, and then do an analysis.
Yeah, I fully agree. So I think we are done now. If you have, what do you think, Jesus? You want to conclude? Or
yeah, I think we came to the end. I want to thank you very much again to Wendy, to you, Jean-Charles, Arne, and Pedro for this, first of all, excellent presentation and discussion. I think we will have many more discussion about this topic in the future for ACC, and this is very good. And just want to remind you that please catch up with the previous journal clubs that you can find in the EASL campus. Thank you very much, everyone, and have a good evening.
Thank you.
Thank you.
Bye-bye.
Thank you.
Thanks.
Thank you. Bye-bye.